Welcome to Unchatted Waters. In this episode, we'll be talking about the microscopic world of pathogens, bacteria, and viruses. But most importantly, how to keep our seafood safe to eat, considering human health now and in the future. So make sure you pop the cork on that champagne, get your oysters ready, as we peel back the shell into this fascinating topic. Welcome to Unchatted Waters, the podcast that talks to the people behind the science. I'm Kirsty, your host, and I'm so excited to introduce my two guests. Craig Baker-Austin is a principal microbiologist focusing on the microbiology of shellfish, and his research interests bridge the gap between marine microbiology, molecular biology, and marine bacteria. Andy Powell is a microbiologist working to make sure our food is safe to eat, and he works with international and national teams to monitor aquatic animal health. Thank you both for joining us. Welcome to the podcast.
Good morning.
Hi.
So first of all, those are quite technical biographies that we've gone through there. So I was wondering if we could summarise a couple of the terms that we mentioned. So Craig, can you explain what microbiology is, please?
Sure. So essentially it's the study of microbes, bacteria, viruses, typically small things that you can't see. Some that are important for human health and some that aren't. Some that play really important roles in ecosystem services, geochemical cycles, things like that. They're really important. Bacteria in particular, which myself and Andy work on, are really important from a food safety perspective.
So the jargon alarm's gone off. Can you explain what an ecosystem service is, please?
Yeah, it's a positive role that something plays in the environment. So with something like bacteria, they play really important roles in things like fixing nitrogen in the carbon cycle. Certain bacteria that are present, for instance, in your body play a really important role. You've got more bacteria in your body than you've got human cells. So actually, it's a really interesting concept. Bacteria have these really important intrinsic roles in almost all of life.
So in your biography, we also talked about molecular biology. So what is the difference between microbiology and molecular biology in this context?
Molecular microbiology, molecular biology is really the study of the molecular basis of different organisms. So it's things like the genetics, the genomics, the proteins that are produced by different things. Study of things like transcriptomics, which is study of genes being expressed. So it's a very diverse subset of microbiology that covers a lot of the detailed and quite tricky basis of molecules and how they interact with an organism.
So there are lots of omics there. So what's the difference between genetics and genomics?
Genetics is the study of the movement of genes, really. Genomics is the study using tools to look at genetic makeup of an organism.
Fascinating. So Andy, we've heard some of those definitions from Craig. What do microbiologists do?
So we look at pathogens like bacteria and viruses that will cause harm to animals or people. There's a lot of lab work involved. Firstly, collecting the sample that could be the vital organs like the liver or pancreas, and then those samples are brought back into the lab where we start our diagnostic work in the laboratory.
So what's some of that diagnostic work? What does that entail?
So it would be a suite of well-defined methods to discover what might be causing the disease in the animal or the pathogen that might be harmful to human health in, say, a food sample such as oysters.
So one thing I wanted to clarify, when we're talking about a pathogen, we are describing something that can cause harm. Is it as separate to virus and bacteria, or viruses and bacteria kind of under the umbrella of a pathogen? Can you define that for me?
A pathogen is a general term for an agent or an organism that can cause a disease or a disease syndrome or manifestation in humans or animals. So they fall under both. There's bacteria and viruses that can do both of those things.
Can we go into a bit more detail about some of the hazards in seafood?
One of the things that's important with seafood is that certain types of seafood are very effective at concentrating different pathogens, that could be bacteria or viruses, from the environment. And really the pathogens can be split into two main groups. Anthropogenic would be types of bacteria and viruses that are introduced into the environment through human activities, and one of the main types of activity would be sewage contamination. The other risk is a naturally occurring one. So there's certain things like harmful algal biotoxins. So these are small algae-like creatures that produce toxins. There's also naturally occurring bacteria that are present in the environment, and under certain conditions they can proliferate, they can grow, and then again they're filtered up and concentrated by shellfish. So there's those two main groups, anthropogenic and naturally occurring.
So we've talked about some of the hazards that might be present in seafood. So what is the effect on seafood if they've got one of these pathogens? A risk to human health, or, should I be eating oysters?
Absolutely. A lot of high quality seafood in the UK. It's a good source of protein. The shellfish industry is very regulated in the UK to ensure public health.
Oysters are a fantastic example of this. So there's a suite of different and quite comprehensive controls that are in place for these types of shellfish species. So one of those would be the E. coli, the microbiological monitoring. That takes place typically monthly, and it's to look for the sanitary conditions in which those shellfish have been grown and then harvested. The other thing that we do in the lab is biotoxin testing. So we look for particular toxins. Some of those can cause problems, and those are monitored very regularly using a range of different methods. And taken together, those two bits, the microbiological and the chemistry testing, really helps us ensure safe seafood.
Brilliant. So let's get into some of those techniques and those methodologies that you talked about there. So can you go through some detection and monitoring methods in a bit more detail?
Yeah, so the micro test for E. coli is typically a wet microbiology technique where you whiz up the whole shellfish and liquor, and then you look for the amount of E. coli bacteria within that food sample.
So when you say liquor, do you mean like you make a shellfish smoothie basically and then put it into your analyses? Can you describe a bit of the analyses in a bit more detail? So once I've got my shellfish smoothie, what happens?
We try and revive the bacteria because they've been in the environment, so they're used to being in the bodies of warm animals. And then after we've revived them, we subculture them onto specific microbiological media to see if that particular bacteria of interest is present and how many are there, which is the important thing.
So there's a lot to unpack there. I'm going to delve deeper into this topic because it's fascinating. Just what is a microbiological media? Can you explain some of that?
Okay, so it's a soup of things that will help revive your bacteria. So they're usually a carbohydrate source and maybe some sugars and an indicator of some kind. Maybe a chromogen in hard media or agar that will show you whether your bacteria is present.
So, sorry, the jag alarm's gone off. What's a chromogen?
So, yeah, it's usually an enzyme that's produced when the bacteria grows, and it will show as a colour on the agar, so it's very clear to the analyst to see.
So basically, in simple terms, you're making a shellfish smoothie, you're then putting it on a nice environment to revive it. You're then putting it on kind of like a piece of paper which it'll light up if it's present. Is that a good analogy?
Yeah.
Great, thank you. So is there anything else that we do in terms of understanding presence and absence of some of these pathogens? For example, you talked about molecular diagnostics. Craig, could you go into a bit more detail of those?
Sure. So one of the things that we do, and we're one of the first laboratory in the world to help develop this method. It's the first virus standard method that's used around the world. It's slightly different to the smoothie that Andy was just talking about, but you take your shellfish and then you remove the digestive gland. Now, the digestive gland is essentially the stomach of the animal, and that's where most of your nasty pathogens or bacteria end up. And we do that because we know that that's a good source of material for the molecular analysis. Pulled together, you normally take, say, 10, 15 oysters. Those are chopped up in a little dish, and then those are extracted. So you're looking for RNA, which is ribonucleic acid, and that's the genetic material that we're looking for. And that can then be detected using a couple of different methods, but one of them is called, and it's a bit of a mouthful, but it's called quantitative real-time PCR. And that's a method that's used in all fields of molecular biology. For instance, during the COVID pandemic, if you had a COVID test, it uses the same type of approach, just uses a different assay at the end. It's a method that's used very widely to detect and quantify different human pathogens such as viruses in samples.
Do you look at any complete sequences? Is it just one little section of DNA you're looking for, or are you looking for the entire kind of genetic makeup of some of these pathogens?
That's an absolutely fantastic question. Yes, we do. So for the detection methods, they typically use a small fragment of either a bacterial or a viral target. So with something like norovirus, you've got a small part of the virus that's found in all norovirus strains, and that's what you're trying to detect. And those are typically called species-specific markers. It's actually the same what's used for COVID. So most of the PCR tests that are used to detect COVID would be focusing on the spike protein, which is highly conserved, and so that's used to be able to detect that you've actually got that pathogen in your body using these molecular methods. Now, where we do things slightly differently is we do also try and sequence the whole thing. That's, I would say, a little bit easier with bacteria. So in the types of work that I do, I'm really a bacteriologist. You get the strains from samples quite easily, and then you can extract the DNA and you can sequence the whole genome really, really quickly and easily now. With viruses, it's a little bit trickier because certain viruses, for example, norovirus, they're quite difficult to grow, and I don't think anyone can really reproducibly grow noroviruses from shellfish samples. So what you're having to do there is to use slightly more complex methods to try and get the whole genome. And that field of molecular biology is called metagenomics. So that's where you're trying to sample and analyze everything in a sample using sequencing-based methods. And then after, it's a little bit like getting a jigsaw puzzle and throwing it up in the air and then trying to put it all back together again. It's really complex. It uses a lot of different, what are called bioinformatic methods. So these are approaches to be able to understand short and sometimes long fragments of DNA and RNA and to be able to put them back together again in a meaningful sense.
Fascinating jigsaw puzzle. Genetics is quite a well-known method throughout science. Is there anything that we're developing, Andy, that is more research-based that might be a little bit different to those genetic approaches?
Yes, we're always looking for ways as scientists to make the methodologies quicker, so you can have a more rapid response to an outbreak or to finding out what pathogen is there. So that's a really important thing that we do in the laboratory.
So one of the things we've invested in is what's called a mass spec machine. It's a machine that you can use to identify bacteria really, really quickly. So to give you some context, it normally takes 3, 4, 5 days to do our normal microbiological tests in the lab, depending on what they are. So for getting an environmental sample or a strain and then going all the way through to the end for the identification, it takes that sort of length of time. This machine is fantastic because what you can do is you can load samples into a specific plate, it gets loaded in, and then the samples are basically blasted open using a laser and then fired along this chamber and then detected at the end. And each different bacterial species has a different fingerprint. I won't go into the many more of the details of how that actually works, but essentially you've got then a database with all these fingerprints in it, and then you can put your unknown sample in, and within, I think, 30 seconds to a minute, you get a result back. So to put that in some context, you can analyse dozens of samples really, really quickly. Whereas before, to do the same test, it would be several days. So it's been a massive step change in what we can do in the lab in terms of identifying bacteria from the sample.
So can you do that for all of your samples moving forward, or are you restricted to a few that are kind of already got their fingerprint mapped?
I think most of the bacteria that we're interested in, Andy, you can interject here, most of the ones that are human pathogens are on the database, I think, and then you can match those against what's present in your sample. And the real utility here is the speed and how high throughput it is. And these are now being used actually in a lot of clinical laboratories, at hospital laboratories. So if you have an infection and then they take a clinical sample, it goes into the lab. That would typically take several days to get a result. Now they can identify it far quicker. And that's important because if you need to treat somebody with an antibiotic, you really need to know what infection they've got. You can't really second guess something like that. Getting a result back in minutes means that we can actually do things far more quickly, and we can make decisions on the ground. And that's got impacts not just for in a clinical setting, but in the types of work that we do, where we're testing samples from environmental sources and also some clinical samples as well. It means you can get an idea of what's going on much, much quicker. So although it was an expensive machine to buy and to invest in, I think it's really going to revolutionise what we can do in the lab.
So we've talked a little bit about the innovation in technology. Is there anything that we are researching at the moment that is going to help with keeping our seafood safe?
So yeah, one of the things we're really interested in here in the laboratory is what are the types of antimicrobial resistance that is in the environment. So antimicrobial resistance is a natural phenomenon we find in bacteria that they use as a survival strategy. Now, we are interested in antimicrobial resistance. We'll call this AMR from now on, because it's been coined the silent pandemic by the United Nations, and every country has been mandated to have a national action plan where they look at AMR in the environment. So this is a real area of research at the moment. We know a lot about what's happening in the clinical environment, but less so in the natural environment. So that's something we're really interested in here in the laboratory.
I go on from there, Andy, that one of the things we're also interested in is how risks change over time. So in microbiology, things never stay the same. It's always in dynamic flux. There's new things emerging all the time. And because of changes to things such as climate change, we're now starting to see different types of risks emerge. And I think it's really important that we keep an eye on that and that we have an understanding about what's going on. So there's a couple of examples. One of those would be a toxin. There's one called TTX, which is tetrodotoxin. It's a really fascinating toxin. It's a very potent neurotoxin. And in fact, it's found in pufferfish, which is a delicacy in Japan. And it's something that we've detected in Europe in the last few years. And having the methods to be able to keep an eye on that particular toxin and its presence is really important. Another thing that I work on and that I lead in the lab is looking at environmental pathogens associated with climate change, in particular bacteria like Vibrios. These are bacteria that grow in warm water, and their abundance in the natural environment tends to track environmental temperatures. So when it gets warm, they proliferate very rapidly, and then they're found in shellfish in high concentrations during warmer months. Those types of bacteria can cause infections in humans, and we tend to see a big increase of these cases during the summer months. Now, traditionally, these bacteria have always been found in places where it is quite warm, or tropical, subtropical environments like the Gulf of Mexico, in the Far East. Now we're starting to see these infections reported in quite colder, temperate areas, the Baltic Sea, certain parts of Western Europe, the northeast coast of the United States, which is typically quite a cold environment. These types of bacteria and the infections associated with them are moving in response to climate change. And it's something that we're looking at using a range of different things. So looking at satellite imagery, climate data, oceanography, microbiology, molecular biology, which we've already talked about, epidemiology, a range of different scientific disciplines. So what's really interesting actually is that you can look at these types of impacts using a range of different multidisciplinary approaches.
Sorry, the jargon alarm's gone off there. What's epidemiology, please?
Epidemiology is the study of essentially the spread of diseases. So it's looking at the movement of diseases, for instance, through an environment, through a community. The sort of godfather of epidemiology I was taught at university was a guy called Dr. John Snow. He did one of the most basic sampling strategies and the first ever type of map of its kind. It's really fascinating. There was a big outbreak of cholera in London in the early 1850s. John Snow was a famous physician, but he was interested in what was causing this cholera, why was it moving around, where was the reservoirs, what was going on. And what he actually found was he mapped all of the cases in central London, and he noticed that they were all clustered around a water pump. Now what's fascinating here was up until that point, a lot of people thought that cholera was transmitted via dirty air, particles in the air, and that's not the case. It's a waterborne pathogen. So once he mapped all of these cases in this particular area, and you can find the map online, it's a really famous scientific image, they removed the handle from the water pump and all of the cholera cases vastly reduced. And it was the first demonstration really of public health intervention using epidemiological data, looking at case data where infections have been reported in the community. And things like that are, I think, so powerful.
So you've talked a little bit about risks changing and being dynamic and never staying the same. So how have these risks changed over time, and what have we already seen happen?
So one of the big changes has been in terms of risks associated with seafood and with shellfish. So a long time ago, in the 19th century and early 20th century, one of the main risks associated with eating seafood was typhoid, which is caused by a particular bacterium called Salmonella. And we improved the sanitary conditions of water treatment, we improved public health interventions, and thankfully now typhoid is no longer an endemic infection in the UK. And what I mean by that, it's not something that we regularly see. People do still get typhoid, it's still brought back into the UK, but it's from areas, parts of the world where perhaps they don't have quite a good system of treating sewage or dealing with water quality issues. So that's how something's changed over time.
Brilliant. Thank you for that bit of detail. Now, I think we're coming to a really nice point in the episode where we can think about the future of seafood. What's it going to look like, do you think, in the future?
One of the things that we're interested in with a growing population is that we really need to provide high quality protein for people, and the way that we need to do that is through aquaculture. It's going to be a really important way to feed global population in the future in all countries. So as that industry grows, we need to develop methods that are more rapid, more efficient, that are easy for people to use as well, that are not necessarily experts. Using COVID as an example again, how quickly the detection of COVID was rolled out to many different labs across the globe.
Craig, did you want to jump in to kind of tell us what you think the future of seafood safety looks like? Is there any kind of technology that you've had your eye on in the bacterial world?
Yeah, sure. So, I mean, Andy made some really valid points there. We've got a growing global population. It's going to need to be fed in the future. And I think we've got a realisation now, we're going to have to move to looking at aquaculture. And it's a big growth area at the moment. What I'm really excited about, whole genome sequencing, which has just been revolutionised in the last few years. So to put it in some context, a long time ago when I did my PhD, which was 20 odd years ago now, it used to cost hundreds of thousands of pounds to sequence a microorganism, just one single bacterium. Now it costs probably 30, 40, 50 pounds to do the same sort of thing. So the costs and the speed associated with it changed rapidly in the last few years. Now, although that's amazing, the problem we have now is about the data that's generated, because you can generate vast amounts of data very, very quickly. And it's how we can use that data in a meaningful sense. So that's really where the main challenge has been, is getting those bioinformatic tools that I mentioned earlier on, and using those in a means to be able to work out characteristics, different aspects of a certain microbe. COVID, and we keep going back to COVID, is a really fascinating example, but when that particular pathogen was identified by the World Health Organization, I think it was sequenced in less than a week. And to put that in context, other pathogens that have caused global pandemics, it took sometimes months or even years before we were able to work out what was going on. And what I'm really excited about now is how we can share data far more readily between researchers in different countries. And that's something that's come out of the advances in molecular biology over the last few years.
Amazing. I'm really trying to understand how much DNA do bacteria have compared to humans? Because, you think about sequencing a genome, that's something that many people might have learnt at school. The amount of genetic material they can transfer between each other, does that mean that they've got thousands and thousands of genes to map? Or could you go into a bit more detail?
Yes, so a standard microbial genome is generally somewhere in the region of about 3 to 6 million bases in length. It's a fraction of the size of the human genome.
So where do we store all this data? You've said that there's a huge amount of data to then process and have as technology gets faster, providing more data. Do we have somewhere to store that everyone can access? And do you think there are greater opportunities in the sector to explore more and more things because we have so much data?
Yeah, so what we generally do, once you sequence a microorganism, and once it's been analysed, that data can then be uploaded from your PC, from your computer, onto a global repository. And one of those is called NCBI, which is a big global repository which has got lots of different microorganisms on it. And there's other examples. That's really one of the challenges, is actually some of the data storage associated with these sorts of projects, because they do take up a lot of space.
We're getting to the end of our episode now, and I think it'd be really good to go into our fun facts. This is our feature where we have a topic or a fun piece of information that our listeners can take away to their family or friends or at a party as that conversational opener. So I'm going to start with you, Andy. What's your kind of fun fact?
My fun fact is that across the UK we have a whole bunch of reference laboratories, particularly designed to answer questions around certain pathogens. And then that spreads out internationally to other global laboratories. So behind the scenes, there's lots of people working on answering the questions that are the hot topics. This is a satellite laboratory where we can go and help lower and middle income countries to help build their capacity for aquaculture, disease control, biosecurity, for humanity, really.
Thank you. So now on to you, Craig. What is your fun fact that people can take away from listening to this podcast?
Okay, so mine is that one of the bacteria that I work on, and probably my favourite bacterium, is called Vibrio vulnificus, and it causes infections, but it's a really fascinating organism because it affects men far more than women. And in fact, we've looked at quite a lot of long-term data now, and it's about 85% of cases are in men and about 15% cases are in women. And that's really strange to have a gender disparity with a particular human pathogen. There's been some studies that suggest that estrogen plays a role in protecting you from some of the toxins that this bacterium produces.
Thank you for your time today. It's been an absolutely fascinating topic, and loved delving into the science lab and learning all about microbiology, molecular biology, bacteria, viruses and pathogens. And thank you both for your time. Hope you have a great day.
Thank you.
Thank you very much.
Thank you for listening. This podcast was made by CEFAS, the Centre for Environment, Fisheries and Aquaculture Science. To keep up to date with us, follow us on social media using @CEFASGovUK. We hope you can join us again next time.